My name is Mark Well. My project consists in developing a gene editing analysis pipeline in a sparkR. So basically, the motivation is to produce an efficient method to analyze the gene editing experiments, as I said. These experiments usually produce large data sets of text files that I think they will be processed very efficiently with this tool. So basically, the decision of using a sparkR is for two reasons. It's because basically it combines the very big abundance of genomics software libraries that R has available, especially through the bioconductor repository, with a Spark large capacity for big data. Very briefly, I'm going to explain how is the process of a gene editing experiment. Basically, we start with treating the cells with a reagent to modify one specific part of the genome. Then we use PCR to basically amplify this loci that we are planning to modify. We assume that this PCR product is representative of the population of edited cells. And then what we do is we take this PCR product and we send for Illumina sequencing. The sequence obtained, basically the Illumina sequencer provides with a very large amount of sequences which represent the different sequences present into the edited loci of the cells. And what we do then is to compare the sequences to the reference loci to analyze what was the efficiency of our editing and how we change these cells. So this is a typical example of results where in the x-axis here I'm showing the position of the reference, and in the y-axis the number of changes. So for example, in this case, most of the changes were located in this area. Here is deconvoluting in either deletions, in this case, or insertions. And here in these two plots, I'm showing what are the distributions of deletions and insertions. For example, here, most of the deletions are one base pair. The same with insertions. So what are the current challenges with the pipelines that exist? So that, as I said before, the next generation files are very large and the current execution is low. So I think that an implementation in sparkR may improve a lot this pipeline. So in order to carry out this project, so basically I had to pre-install many different packages of software. So the sequences that come from the sequencer always come in two sets, read 1 and read 2, because each cluster of DNA is read from one side and from the other. So we need to use this tool pair to merge the two reads. So basically, since they both overlap, we can use some computational tools to merge them. Then the second very important software is the mapper. So basically we get this list of sequences, then we need to basically compare them to the reference for getting the coordinates of where they map and also to get the differences. So the coordinates is a number, and basically the difference is encoded in what is called a sparkR field, which is this one. In this case, it would be a 151 match. Then in addition, we need other software installation. So we need a couple of bioconductor libraries. One is called Genomic Alignment, and the other one is RSAM tools. Both libraries will allow us to load and explore the genomic data in R. And then another library is SAMtools, which will allow us to manipulate the different objects that we will generate. And very important, I had also to install sparkR. So I basically downloaded from this link, and I loaded, and basically I loaded the Spark workspace into R. So some more details of the original data. As I said, the biggest amount of data comes from what we call the FASTQ files that contain all the sequences that we sequence from the populations of cells. And these files are very simple. As you can see, are very long text files. In the case that I am using will be a 30,000. Normally they are much bigger. 30,000 reads, normally we have millions of reads. And basically they always consist of a line that encodes the sequence and then, an identifier for the sequence, a plus symbol, and then some quality value that informs of how well was sequenced each base. And then the other part is the reference file, which is a very small, in this case, text file with the sequence of the loci that we are trying to modify. Okay, so then how do we prepare the data? First, I need to merge the two reads. As I said, we have reads from one side and from the other. I generated a single read. Then I need to map these reads to the reference. So first I generate an index from the reference, and then I generate alignment. And finally, I also need to do some pre-processing to later on visualize the results. And basically, this processing is obtaining a smaller alignment object that basically only contains the mutated sequences, because this will be the most interesting to visualize later on. Here is how I load Spark into R. So basically, I need to indicate to R where is where is located the Spark in my laptop, and then basically I load the workspace. So next step is to load the data once we started R and the Spark session. So to load the data, we take advantage of the R libraries that can load this genomics data. And here I'm using the library R SumTools. And then I am loading, basically using this function, the bound file, which is the file that contains all the information of where all the reads map and which are the differences between this map read and the reference. As a second step, what I do is I transform this R object into a Spark data frame, basically only taking the important information for our analysis, which is the position of the mapping, and then the SIGAR term that, as I said before, this SIGAR term encodes the difference between the map read and the reference read. So here is when we use the full power of a Spark. Basically what I do is I summarize the data, this large data set of more than 30,000 in a pilot. In a normal data set would be in the order of millions, into a very small summary of how many types of reads we observe and how many of these types of differences. So basically, this is what we collect from a Spark. This is the table that contains the SIGAR terms. So basically the differences between a certain read and the reference, where is the position, and how many of those. This is basically the result that we were interested in. And then finally what I do is I use the package genome alignments to analyze the differences between the references and the reads so that we can represent them. And as a final step, what I do is I arrange all this data on a convenient way to represent, which is this table that I'm showing here, where we have each position of the reference, position 200, 201, 202, and basically the number of substitutions, insertions, or deletions that we see in each of these positions. So I first generate some summary statistics, which I present here in R. So in this first pie plot, basically I'm just showing the percentage of modified reads, which is about 1%. Here where the deletions are located, which is more or less in the middle of the reference, as expected. Here where the insertions are located, and here I'm just showing two histograms of the sizes, of the distribution of sizes of deletions and insertions. In order to provide more information, I also added another visualization tool, a more dynamic one in JavaScript. So basically, I made use of a library developed in the Jeff Hammerbach laboratory in Mount Sinai in New York. That basically, and he developed basically a genome browser in JavaScript that can be used from the web browser. And what I did is I modified the code of this library to represent my data in this project. So basically, I can show briefly. So here I have the static representations on the top of the website, which include the summary statistics, but then I include this other more interactive area where we can browse the reference, we can zoom in, zoom out, and then we can also, for example, interrogate specific grids, for example, and we get all the information associated with this specific grid. So yeah, and here it's just like another slide with the same. So basically, we can not only see the summary statistics, but also browse exactly how all the data looks like. And then finally, just a couple of conclusions. So what I did is I implemented a new gene editing analysis pipeline based on a sparkR. This increased a lot the processing velocity compared to the classic methods. And then the other big advantage is that many times these experiments are done in big numbers. So with this pipeline, I will have built a much more scalable system for large and diverse datasets. Yeah, thank you very much.